What's up, guys? So today we're going to talk about important monosaccharides, important disaccharides, and some monosaccharide derivatives. So let's start with our monosaccharide derivatives. So we're going to divide these into four major classes: deoxy sugars, amino sugars, sugar alcohols, and carboxylic acid sugars. And we can subdivide the carboxylic acid sugars into aldonic acids and uronic acids. Okay, so what I have here is a Fisher projection for the sugar glucose, which we're going to talk about in a little more depth later today. And since the hydroxy group attached to our last chiral carbon is pointing to the right, this would be D-glucose. Okay, so let's start with deoxy sugars, and let's form a deoxy sugar from our D-glucose. A deoxy sugar is where you replace one or more hydroxy groups on a sugar with a hydrogen. So for instance, we could replace the hydroxy group at carbon 2 with a hydrogen, and that would give us this molecule, which we would call D2-deoxyglucose. Okay, so let's move on to amino sugars. So an amino sugar is where one or more hydroxy groups on a sugar has been replaced with an amine, an amino group. So let's take our D-glucose and replace the hydroxy group on carbon 4 with NH2. And we'd produce this molecule here, which is known as D4-glucosamine. Okay, our next group are the sugar alcohols, and a sugar alcohol is where a carbonyl on a sugar has been reduced to an alcohol. So the only way we could do that with our D-glucose is to reduce the carbonyl at position 1. And that's going to form this molecule right here, which is known as sorbitol. Unfortunately, a lot of these sugar alcohols don't have very intuitive names, but fortunately for you, you don't have to know any of these names. All I'm going to ask you to do on an exam is draw me an example of one of these derivatives given a sugar to start with. Okay, and our next group are the aldonic acids. So an aldonic acid is a carboxylic acid that's been formed by oxidation of the aldehyde group. So if we were to oxidize the aldehyde group on D-glucose, we would get a molecule that looks like this, which is called D-gluconic acid. And our last group is the uronic acids. So a uronic acid is formed by oxidizing the primary alcohol on a sugar. We get this molecule right here, which is referred to as D-glucuronic acid. Okay, so now let's talk about our important monosaccharides. And we're going to start with the one that is most important in you, and that is glucose. Glucose is also sometimes called dextrose, corn sugar, grape sugar, or blood sugar. So the concentration of glucose in the blood is very important for our normal body function, and it's carefully controlled by a pair of peptide hormones known as insulin and glucagon. And we're going to talk about these a little bit more when we get to the proteins modules. So the normal blood glucose levels are typically below 100 milligrams of glucose per 100 milliliters of blood. And this is typically going to be at its highest after we eat. And that's where insulin is going to come in and stimulate the uptake of glucose from the bloodstream to other cells in our body. However, if your blood sugar concentration gets too low and you start feeling a little lightheaded and shaky, this other peptide hormone, glucagon, is going to stimulate the liver to release glucose into the bloodstream so we can get back to normal levels. And our next monosaccharide is fructose, which is also known as levulose or fruit sugar. And this is the sweetest sugar we're going to talk about today. However, it's not the sweetest compound in the world. There are quite a few other compounds that are much, much sweeter than fructose. And you're going to read about a few of those in one of the health links for this chapter. So fructose is found in large amounts in corn syrup and honey. And since it's got the name fruit sugar, as you might have guessed, it's also found in very large amounts in sweeter fruits. Okay, our next two monosaccharides are ribose and 2-deoxyribose. So ribose is a component of ribonucleic acids, or RNA, and some coenzymes that are required by many of the enzymes that carry out the biochemical reactions in our body. So 2-deoxyribose, which has the same structure as ribose, except that we have substituted a hydrogen for the hydroxy group at carbon 2, is a component of deoxyribonucleic acids, or DNA, which is the molecule responsible for carrying the genetic information in our cells. And the last monosaccharide we're going to talk about today is galactose. So galactose, along with a galactose derivative, beta-D-N-acetylgalactosamine, are components of blood group antigens that determine our blood type. So let's take a look at the structure of galactose and N-acetylgalactosamine. So acetyl is a name that you may not have seen before. So an acetyl group is this structure right here, where you have a carbonyl attached to a methyl group. So that means N-acetylgalactosamine is a galactose that's been converted into an amino sugar, and then we formed an amide by attaching an acetyl group at position N. And another reason galactose is important is because along with glucose, it makes up one of the most important disaccharides, lactose. So lactose is formed from a galactose residue and a glucose residue, and it's going to be the principal sugar found in milk. And our last disaccharide is sucrose, which is also known as table sugar, cane sugar, beet sugar. And if you ask anybody outside of science and they say sugar, they're probably talking about sucrose. So sucrose is a disaccharide made up of a glucose residue and a fructose residue. And this is an important sugar that's synthesized by plants, but cannot be synthesized by animals. So what do us animals use sucrose for? Well, one way is a preservative. So high concentrations of sucrose can cause high osmotic pressure, which is going to inhibit the growth of bacteria. But more commonly, it's used as a sweetener. In fact, the average adult in this country consumes over 150 pounds of sucrose per year. Okay, so now that we've talked about these important monosaccharides, let's do a little review here. So let's do some reactions with monosaccharides. So this is nothing new. This is a reaction that you saw in the last module. It's just instead of working with a small aldehyde this time, we're working with a larger biomolecule, a monosaccharide. So I want you to pause this video. I want you to draw the reactant, identify if the reactant is being reduced or oxidized, draw the product, and identify the major class of the product. So either a deoxy sugar, amino sugar, sugar alcohol, aldonic acid, or uronic acid. Okay, let's see what you came up with. So this is a reduction reaction. So I'll start by drawing our sugar, and we're going to react this with H2 in the presence of a metal catalyst. And we're just going to break our double bond between the carbon and the oxygen. And we're going to add an H to each previously double bonded atom. So we have created a sugar alcohol. Okay, let's try another one. So same thing, draw the reactant, identify whether the reactant is being reduced or oxidized, then draw the product and identify the major class of the product. Okay, let's see what you came up with. So this is an oxidation reaction. So I'm going to start by drawing our reactant, and we're going to react this with Benedict's reagent. And remember, Benedict's reagent can only oxidize aldehydes, so we end up with the conjugate base of an aldonic acid. Okay, so speaking of Benedict's reagent, let's talk about reducing sugars. So a reducing sugar is any sugar that gives a positive Benedict's test. So that means any sugar that has an aldehyde, because that aldehyde is going to get oxidized to a carboxylic acid, while copper 2+ is reduced to copper 1+, and that's going to cause a color change from blue to orange. So I want you to remember, we're going to draw most of these sugars in their cyclic hemiacetal form. But because of mutarotation, they're going to open up and spend some amount of time in solution as the aldehyde form. And when they open up to that linear aldehyde form, they're going to be able to react with the Benedict's reagent. So that means when you're trying to determine if a sugar is a reducing sugar or not, you need to check to see if it has an aldehyde or a hemiacetal group that can open up to form an aldehyde. Okay, so now I'm going to throw a curveball at you. So fructose is actually a reducing sugar. So go ahead and look back at your notes from this video and classify fructose for me. So classify fructose by size and functional group. So that's right, fructose is a ketohexose. So fructose does not have an aldehyde group, whether it's in the linear form or the cyclic form. So fructose is a reducing sugar because of something called tautomerization. So tautomerization is an isomerization process. So this is where some constitutional isomers can readily interconvert under certain conditions. So under the basic conditions of the Benedict's test, fructose can undergo what is called an ene-diol reaction. And it's called that because it forms this intermediate where you have a carbon-carbon double bond, hence the name ene, where both carbons are attached to hydroxy groups, diol. And just like the enols that we talked about earlier, ene-diols are also unstable, so they're going to convert back into fructose or into glucose. So if the ene-diol converts into glucose, glucose has an aldehyde, so that's going to be able to interact with the Benedict's reagent, and so we're going to get a positive Benedict's test. So this is why fructose is a reducing sugar, even though it does not contain an aldehyde group. Okay, so what about disaccharides? Can they act as reducing sugars? So let's take a look at the molecule on this page. So it doesn't have an aldehyde, but does it have a hemiacetal group that could open up to form an aldehyde? So this hemiacetal group on this rightmost glucose residue here can open up, form an aldehyde, and then it can react with Benedict's reagent, so this disaccharide would be a reducing sugar. So let's try one more example. So what about this disaccharide? So this is sucrose, which we talked about earlier. So is sucrose a reducing sugar? So sucrose does not have an aldehyde, but does it have a free hemiacetal group that can open up to form an aldehyde? And the answer is no. There is nowhere on this molecule where you see the HOCOC linkage. So this molecule cannot open up to form an aldehyde. So sucrose is not a reducing sugar.